Hello everyone, this video is about the key terms in experimental design. So we're going to cover these 21 keywords. If you don't have a note of them already in your independent study, I'd suggest you add them to your notes. Okay, so the first two are very straightforward. These are basically the variables that you guys know and use very regularly in your practicals. So just a reminder that the independent variable is the variable that you change and you're in control of. The dependent variable is the one that you measure. And just a reminder that with these, you always put the independent variable on the x-axis and the dependent variable on the y-axis when plotting graphs. Controlled variables, so please do describe these as controlled variables, not just control variables. And these are basically what you keep consistent throughout a practical to make sure that the only thing impacting the dependent variable, whatever you're measuring, is the independent variable. Okay, now this ensures that you have a valid experiment. So controlled variables are very important to ensure you have a valid experiment. And let's take this experiment here, for example. So this is an experiment to measure the production of oxygen in photosynthesis of this pondweed. And let's imagine that our independent variable is going to be distance. And we're measuring the volume of oxygen in a certain time period, like 30 seconds or something like that. So there are a few controlled variables linked in with this. And when we're talking about this, for example, in exam questions, we want to make sure that we're as specific as possible. So things that could affect the volume of oxygen produced or the rate of photosynthesis will be things like temperature. Photosynthesis is a series of reactions controlled by lots and lots of enzymes, and as you guys know, an increase in temperature increases the kinetic energy and therefore increases the rate of reaction. So we might use something like a thermostatically controlled water bath to carry out this experiment in, okay? And that would mean that we'd have some sort of thermostat that would then regulate the temperature, whatever temperature that we wanted it to maintain it at, the optimum temperature. We might monitor the temperature using a thermometer, but we would regulate it using a thermostatically controlled water bath. So that would be one controlled variable. There are lots of others, so things like potentially the surface area of the leaves found on the pondweed, that might be another one. Maybe the pH of the solution, so here we're using water, but is it deionized water? Is there anything else present in it? Also maybe the type of light bulb. So again, when you're talking about controlled variables, try to be as specific as possible, especially at A level. And when you're describing ways of regulating those controlled variables, again, be really specific. I couldn't just say use a thermometer, because a thermometer doesn't regulate the temperature of that solution that the pondweed's in. I'd have to say a thermostatically controlled water bath, because that would use the thermostat to regulate the temperature of that solution. Okay, these key terms regularly get used in the wrong way and sort of confused amongst themselves. So the first one is accuracy. Now this is basically how close to the actual value is your measurement. So let's say I'm measuring the length of something like a plant leaf, okay, and here's my leaf and here's my ruler, and let's imagine here that we've got inches. Okay, so there's my unit, inches. Now, if I'm measuring my leaf to the nearest inch, that leaves quite a big percentage error range. So a better thing for me to do to get a more accurate reading would be to use a smaller unit. Okay, so something like millimeters rather than inches would give me a more accurate reading, closer to the true value of that leaf. Millimeters are more precise as a unit, meaning they're smaller as a unit compared to inches. Okay, so often if you're asked to suggest how you could maybe improve the accuracy of an experiment, you really need to think about how you could use smaller intervals. So for example, down here, rather than using like, let's say pHs, 0, 2, 4, 6, 8, we might use smaller intervals, so 0, 1, 2, 3, 4, 5, 6, 7, 8. Okay, for example. Validity, so how well does the experiment test what it's meant to test? So this is basically, is the only thing affecting what you're reading or what you're measuring, the dependent variable, is the only thing affecting that the independent variable? Is there something else that might be affecting your reading? So to ensure that your experiment is really valid, you have to basically make sure that you've identified all controlled variables. Again, as we were just saying in the last slide, so give examples as well of these controlled variables and try and be as specific as possible with how you're controlling them. We might also carry out a control experiment. So control experiments, please remember, are different controlled variables, hence why we have to be careful with our phrasing there. So a control experiment is generally where we are removing the independent variable. Okay, control experiments sometimes, so for example in exam questions you might be asked to suggest a control experiment, so sometimes they are quite hard to kind of identify. General rule of thumb is just think about how could I remove or inactivate the independent variable. Okay, now a good example of this was when we did the enzyme catalase practical earlier on in the year. So your control experiment for that practical was to boil the enzyme and therefore denature it. And then when you carry out the experiment, nothing happens because the enzyme's denatured. So there we would be removing the independent variable by boiling it. Another thing that you can do to improve validity is to increase your sample size. So a good example for this would be, let's say I wanted to carry out a survey on all of the students or on a sample of students from college. It wouldn't be a very valid survey if I was to just ask like 5 to 10 people. That's not going to be representative of the student cohort as a whole. So a better thing to do would be for me to maybe ask 30 to 50% of the cohort. So that would be what, like 1,000 to 1,500 students kind of. So much larger sample size, and that would be a much more valid way of measuring. Reliability, so this is how much can you trust your data. And the easy way to check reliability is to basically carry out repeats. So you'll carry out repeat, you'll check that you get similar data each time. You can also calculate a mean and then identify outliers as well. So just to kind of touch upon how these link in with graphs and a couple of other images here. So with reliability, if we start with that, now you guys often plot graphs, so you'll recognise a lot of these. So here we've got a line chart at the top, and what we can see is that we've got two different measurements, and this is just arbitrary data that we've just grabbed off Google. So what we can also see here is the means are plotted as a little dot, and then we've got these little I bars here. So these are basically, different things can be plotted there. So you might plot the standard deviation as this bar. You might plot the range, so you might plot a range bar, or you might plot the error, percentage error. Okay, sorry, excuse my writing, but what we can notice is, for example, if we're plotting the range, then we can see the bigger the bar, the more scattered that data set is. So when I carried out my repeat experiments, the data didn't come back the same every time. If it comes back the same every time, you would just have a data set kind of like this, so where there's just no range at all. Okay, and that's very reliable data. So here at the bottom, that's a very reliable data piece or data set. Whereas over here, for the same whatever this blue line is, we can see that the data is more spread out, so therefore that's a less reliable data set. And here we could actually compare the two. So let's say, for example, it's an experiment looking at the rate of reaction at different pHs or something. So we can see, the blue pH, for example, we might compare the reliability of that data set with the red line that maybe represents another pH. And you can do it similarly with a bar chart as well. So here you can see we plot the mean as the bar as a whole, and then the range, standard deviation, percentage error, whatever it is you're plotting, as that little I bar. And again, the bigger the bar, the less reliable the data because it's more spread out. The smaller the bar, the more reliable the data. One other thing just to think about here is, does the data overlap? So let's say we were taking 3 measurements of, I don't know, organism X, organism Y and organism Z. And we wanted to see if a factor affected those organisms differently. Now, one thing that we can also spot here is how much these bars overlap. So let's imagine that this is here, these bars plotted are the range. We can see that some of the data collected for organism X overlaps with some of the data collected for organism Y, and some of the data collected for organism Y overlaps with some of the data collected for organism Z. So what that suggests to us is that actually we might need to modify and sort of carry out our experiment again, but just from this data, you know, that data is not looking significantly different. It's not looking, because they're sharing so many data sets, it's not looking like we could say that organism X is significantly higher than organism Y. So it's always important to look at those range bars as well, rather than just looking at the means, because, you know, the means, these bars as a whole, they do look quite different, but then when you actually look at the range bars, it says a different story. Another way of describing accuracy, so again, lots of people do get confused with this stuff. So if we're just to look at accuracy, now let's imagine down here, so I'm wanting to fire my, I don't know, Nerf gun, whatever, at the target, and I'm aiming for the centre, I'm aiming for the bullseye. So here, if I hit the bullseye, as in this example, I'm being accurate with my aim. So I'm getting it as close to the thing as close to the true value, or as close to the real thing that I'm aiming for. Whereas over here, I'm not very accurate because I've missed the bullseye. One thing that we can, though, look at here is that the reliability of my shots is actually quite good. So in this first example, I'm accurate because I'm hitting the bullseye, and I'm reliable because I'm getting them in the same place each time. Whereas over here, on the second one, I'm not accurate because I'm not hitting the bullseye, but I am reliable. You know, every time I'm missing in pretty much the same place. Okay. So the difference between causation and correlation, now this is something that we just as scientists have to be quite wary, and I think actually most of you are probably very, very good at assessing for this anyway. But just because something's correlated, it doesn't mean that one thing causes the other. Okay, so a good example here is to look at something like shoe size compared to IQ. Now, this is a genuine correlation. As shoe size increases, so as one variable increases, so does the other variable. And you do see that if you were to analyse the data. But that doesn't mean that if you've got massive feet, you've necessarily got a really high IQ or vice versa. So just maybe pause the video and just have a think about why that correlation exists. Okay. And some of you might have spotted already, but this correlation is because it includes children. Okay, so kids have very, very small feet, and they're also learning and developing. So they haven't, you know, fully developed the skills to answer an adult IQ test in the right way. So just to bear in mind that, again, you might see a correlation in data, but that doesn't necessarily mean that one variable causes a change in the other one. So the difference between a null and alternative hypothesis. So for this, some of you might be very, very familiar with this because you might have already done it in a different subject. But if you haven't come across it before, and even if you have for biology, it might help to hear it again. So when you're developing hypotheses for a particular observation or question that you have about biology, at GCSE, you would, and in your first year, you often consider alternative hypotheses. So, you know, when you would originally write a hypothesis as a GCSE student, this is actually what you're writing. Now, for A-level, you need to be able to write a null hypothesis. So think N for null, and N for no, basically what a null hypothesis always says is that there's no significant difference or correlation, depending on what you're looking for, between or observed, and any observations of difference are going to be due to sampling or experimental errors. Okay, so it's basically saying there's no significant difference or correlation in your data. Now, your null hypothesis is the thing that you are testing for in your stats tests, and we'll come back to those next week. But you always do need to write null hypotheses for your experiments, and it's good to just practice them. So you can literally just phrase them like this, but do just be wary and really think about, are you looking for a difference or are you looking for a correlation? Okay. And generally speaking, again, we'll come back to that a little bit more next week as well, but a difference is you're sort of comparing like two categories, and a correlation is maybe your independent variable is spread along a continuous range. Okay, so I don't know, like height or something like that. Your alternative hypothesis, so this states that there is a significant difference or correlation between your two variables. Okay, so again, this is what you guys are probably much more familiar with writing. Okay, so just on that note, if we were to just talk a little bit more about alternative hypotheses. Now, when you're writing these, there's a couple of different ways that you can write them, and developing your alternative hypotheses, if you use a one or a two-tailed hypothesis type, that will basically determine how your stats test is carried out. The reason being is that because it's statistically sort of calculated that if you're a bit more confident in sort of your hypothesis, you're basically able to develop a one-tailed directional hypothesis. I'll come to this in a second, and therefore your statistical testing, the data that you compare it to, is a little bit different. So a two-tailed non-directional hypothesis. Now this is basically where you're saying something like, I think there's going to be a significant correlation between my independent and dependent variable, but I don't know which way. I don't know if it's going to be a positive or a negative correlation, but I think there will be a correlation of some sort. A directional hypothesis, so these are generally a bit better to form. So this is where, maybe through other reading or research, you've identified, well, actually, yeah, do you know this past experiment that I read about, they found quite a similar thing, and from my reading and my research, I think that actually it's going to be a positive correlation between my IV and my DV. So that would be what we call a one-tailed directional hypothesis. You guys will be practicing this much more next week, but Spearmans Rank is one of the stats tests that you need to know for our course. So basically the Spearmans Rank, what we use this for is to check if there is a statistically significant correlation between your IV and your DV. And what you'll get from a Spearmans Rank test is you'll get an RS value between -1 and +1. +1 just indicates that you've got a perfect positive correlation, -1 that you've got a perfect negative correlation between your IV and your DV, and 0, no correlation whatsoever. So again, you guys will need to practice this. It's something that you do need to be able to do, and you also do need to be able to interpret that data as well, but just to give you an idea of when that's used. So again, these are used for looking at correlations, so line charts, generally you'll be plotting for those. The student Ttest, so this is, again, I'd like you, whenever you're thinking of a student Ttest, you can think about bar charts. So here we're looking for a statistically significant difference rather than correlation. So just be careful with your phrasing when you're talking about these two, between two sets of data. So maybe we're looking at enzyme activity, and we've got a control experiment and a treated experiment. And we can carry out the student Ttest because actually, looking at this bar chart here, I can see that the means look, relatively different. If I was to compare the range bars or standard deviation, whatever being plotted here, I can see that there is a little bit of overlap here. So I can't, conclusively just say much just based on that graph. I really need to do some sort of statistics on it. So I could carry out the student's t-test and it will tell me if those two means are significantly different from one another. Finally, standard deviation is something that you guys have done a lot at school, and those of you who've done maths and other science-based subjects will feel a little bit more confident about. But standard deviation, you can sort of think of it as like the average spread of the data, the average range of the data. But basically it's how much your data sets differ from the mean, so how far out are they distributed. So here on the left you can see we've got a small standard deviation, and that's because the normal distribution of this data set is very steep, so there's not much difference or deviation away from the mean. Whereas over here we've got a large standard deviation, that's because we've got a wider spread of the data away from the mean. Okay, thank you.